Welcome back to Biology 1406. This is the first part of the recorded lecture for Chapter 8, Photosynthesis. Okay, so the process we're going to be talking about throughout this chapter is photosynthesis, and this is the process that we're going to use to convert solar energy, or light energy from the sun, into a usable form of chemical energy for the plant. Remember that this process is all about the plant and what the plant needs. Anything that we get from this process is purely a waste product, according to the plant. This process, either directly or indirectly, provides the nourishment for every plant, or for every living organism on the planet. Plants themselves we refer to as autotrophs, and these are organisms that are able to sustain themselves without eating anything. They get all of their energy from their own conversion of light into usable chemical energy. We refer to these organisms as producers. You're going to hear more about them in Biology 1407 when you take that class. And their job is to produce organic molecules, primarily glucose and other carbohydrates, from carbon dioxide and some other inorganic molecules. Almost all of the plants on the planet are photoautotrophs. That means that they use the energy of the sun to make organic molecules. But there are some autotrophs that use other forms of chemicals for this, and we refer to them as chemoautotrophs. So what's going on here is that energy will come in to the chloroplasts, and if you remember, chloroplasts were those specialized structures that we looked at in cell structure that contain the green pigments. And as sunlight comes in, it gets stored as chemical energy in the organic compounds. The sugar that is made in these chloroplasts provides chemical energy for the plant to make organic molecules of the cells. The plants also will store excess sugar as starch, so anytime you're eating a potato or a carrot, even corn and peas, beans, all are starch storage mechanisms that the plants use to store excess sugar. The process, as a byproduct, will produce oxygen that allows for us to breathe and allows for the process of cellular respiration to take place as well. All of the organisms on the planet that do not provide their own food and energy are referred to as heterotrophs. These are organisms that will eat other organic materials in order to get their energy, and we refer to them as consumers. Again, you'll talk more about them in Bio 2. Humans and almost every other heterotroph on the planet depends on photoautotrophs for their food and for their oxygen. Without the plants, basically nothing else on the planet lives. You find photosynthesis in any green organism. So plants, algae, some of the protists, including the euglena, and some cyanobacteria, all are able to do photosynthesis, and they feed themselves that way, but they also provide the basis of energy for all of the other organisms on the planet. Record. So photosynthesis is going to convert light energy into the chemical energy of food using some really specialized photosynthetic cells that we call chloroplasts. The chloroplasts are going to contain some enzymes and some other molecules grouped together inside of a membrane, and this allows for these chemical reactions to proceed in a really efficient manner so that the plants are using their available resources to the best of their abilities. The chloroplasts, if you remember back to the endosymbiont theory, are similar to and likely evolved from photosynthetic bacteria that were engulfed by eukaryotic cells way back. Now, inside of the chloroplasts, or you're going to find the chloroplasts inside of the leaves primarily. These are the major sites of photosynthesis, but you will get some photosynthesis in stems. Pretty much any part of the plant that is green is capable of doing some level of photosynthesis. The green color of the chloroplasts comes from a pigment called chlorophyll. And chlorophyll is the primary pigment in photosynthesis, and we'll talk more about it in detail here in a few minutes. They're found mainly in the cells of the mesophyll, and all you need to know about this at this point is that they are found in the mesophyll. You'll talk more about the structure of leaves and the parts of the leaves when you take Bio 2 next semester. Okay, other structures that we're going to talk about in terms of photosynthesis are things like the stomata. And the stomata, which you see over here, are pore-like structures in the leaves that allow carbon dioxide to enter and oxygen to exit the leaf. We have the thylakoids, which are connected sacs that are found inside of the chloroplast, and you can see them here. These stacks, they look like little stacks of green coins. One of the students referred to it as a stack of green Thin Mint cookies. When they are stacked in columns, we refer to them as a grana or granum. Inside of the chloroplast, you're also going to find the stroma, which is a dense interior fluid, and the stroma is the location where parts of the process of photosynthesis will take place. Okay, so we're back to oxidation reduction reactions here, and we're going to talk about photosynthesis. Now, for exam purposes, this is the reaction that you need to be able to write. You should know that there are 6 carbon dioxide, that there are 12 water, and that there's light energy all coming into this reaction, and that from that reaction we get glucose, 6 oxygen molecules, and 6 water molecules. You should also know that carbon dioxide is reduced and becomes glucose, and water is oxidized and becomes oxygen. As long as you can write out this reaction and label which ones are reduced and which ones are oxidized, then you're good to go for the exam in this regard. So photosynthesis is going to take place in 2 different parts. The first part is the light reactions. These are going to take place in the thylakoids, those green stacked coins that we talked about. And the most important things that happen in the light reactions are the splitting of water, the releasing of oxygen, the reduction of NADP plus to NADPH, and the generation of ATP from ADP through a process called photophosphorylation. Once we've finished the light reactions, then we'll talk about the Calvin cycle. The Calvin cycle does not take place in the thylakoids. Instead, it takes place in the stroma, that thick liquid that's inside of the chloroplasts. And the main purpose of the Calvin cycle is to form sugar to make that glucose from carbon dioxide molecules using the ATP and the NADPH that we created in the light reactions. We're going to start with a process called carbon fixation that's going to allow us to incorporate carbon dioxide into the organic molecules that we're going to make as we proceed through this process. This is an animation of photosynthesis that is available to you on the original PowerPoint in Blackboard. I encourage you to watch this. And this is just going to walk you through those steps. So we have light coming in and water coming in to our thylakoids inside of the chloroplast. We also have NADP plus and ADP and some phosphorus that's going to come into the light reactions. Once they come in, then we're going to generate ATP, we're going to generate NADPH, and we're going to release oxygen out of the cell, out of the chloroplast, and then eventually out of the leaf and into the atmosphere. This ATP and NADPH are then going to move into the next process that takes place in the Okay, so ATP is going to come into the Calvin cycle, and when it gets when it gets oxidized, it is going to regenerate ADP that will then exit the Calvin cycle and go back to the light reactions. NADPH is going to come into the Calvin cycle. It will also be oxidized, regenerating NADP plus and sending that back to the light reactions so that this process can continue. The electrons that were released from ATP and from NADPH are then going to go through the Calvin cycle, where they will use the carbon dioxide that's coming in from outside in the atmosphere to convert that carbon dioxide into glucose to complete this process. The important thing to remember here is that for the plant's purposes, the goal of photosynthesis is the production of this glucose. So the light reactions are going to convert solar energy to chemical energy. That is the definition of what photosynthesis is supposed to be doing, right? The chemical energy is going to come in an intermediate form at the end of the light reactions in the forms of ATP and NADPH. Just like we talked about with cellular respiration, these are high energy molecules, particularly NADPH, is similar to the NADH that we talked about in cellular respiration, that will carry that electron from the light reactions into the Calvin cycle. Sunlight is all electromagnetic energy. We can also refer to it as electromagnetic radiation, and it works in the same way as all of the other electromagnetic energy on the planet in terms of wavelengths. So when we talk about a wavelength, we're talking about the distance from one peak to the next peak. And so we're talking about the distance from here to here as our wavelength. The distance between these 2 points, the length of that wave, determines the kind of electromagnetic energy that you have. When we talk about the wavelengths that are important in photosynthesis, we're really talking about the visible light spectrum, which runs from about 380 nanometers to about 750 nanometers. The other thing that's important to understand here, and we'll talk about this more in a minute, is that light has this tendency to behave as though it is made up of these discrete individual particles called photons. And the photons will become really important in the light reactions here in a few minutes. So the pigments that we're going to be talking about are substances that absorb light, particularly visible light, and each different pigment has the ability to absorb a different wavelength of light. Not every wavelength is absorbed, and any wavelength that is not absorbed is going to be transmitted. So what that means is that the reason that leaves appear green when you see them during the summer is because chlorophyll is really good at absorbing other colors of light, all of the other colors of light except the green, and green is then transmitted or reflected back from that pigment.
The thylakoid membrane in chloroplasts contains light-absorbing molecules called pigments. Different pigments absorb light of different wavelengths. One pigment, chlorophyll A, absorbs mainly blue-violet and red light. This molecule participates directly in the light reactions. Another molecule, chlorophyll B, absorbs mainly blue and orange light. We do not see these absorbed colors when we look at a leaf. Instead, we see the green wavelengths that are reflected back from the leaf or transmitted through it.
When we look at the absorption spectrum, we're going to pay attention to a couple of things. We're going to pay attention to chlorophyll A, which is this sort of teal blue line down here. We're going to pay attention to chlorophyll B, which is this green line here. And we're going to pay attention to the carotenoids, which is the orange line here. And what you'll see is that for the most part, violet, blue, and red lights work really well for chlorophyll A. And as the primary pigment, this is an important wavelength for chlorophyll A. You'll see that chlorophyll B also picks up some violet, blue, and some indigos, but it moves a little further into the lighter blues and a little farther into the reds. And then you have the carotenoids, and the carotenoids have a unique job that we'll talk about here in a minute, but I want you to notice that they kind of overlap with both the chlorophyll A and chlorophyll B end of the spectrum down in that 400 to 500 nanometer wavelength. Action spectrums relate directly to absorption spectrums. So when you look at the action spectrum, you'll see that the rate of photosynthesis is highest in these areas that match the areas of highest absorption in chlorophyll A and chlorophyll B. And you have this double peak where you get the absorption down in these red areas as well. Chlorophyll A is the main photosynthetic pigment that you need to be concerned about. Chlorophyll B is an accessory pigment that makes the spectrum a little bit broader and allows for a little better usage of the light. And this is purely based on structural differences between these two molecules. The carotenoids, as I mentioned earlier, have a really important job. Their job is to absorb excessive amounts of light that would damage the chlorophyll. So you've got an overlap between A and B that allow this plant to take in quite a bit of light, but it is possible to get too much light. And so the carotenoid's job is to sort of limit the quantity of light that's coming in to avoid damaging the pigments. When the pigments absorb light, they get excited and they move into an unstable state. As they fall back down from this unstable state, they give off photons and they cause something called fluorescence. All you really need to know about this is that the photons are basically giving off an energy or an afterglow as a result of this.